and Ikbkb mutation. Deficiency of innate and acquired immunity caused by Ikbkb mutation. A form of human SCED is characterized by normal lymphocyte development despite a loss of EK2 function. EK2 deficit results in an impaired response to activation stimuli in a variety of immune cells, leading to clinical relevant impairment of adaptive and innate immunity. Background: severe combined immunodeficiency (SCED) comprises a heterogeneous group of hereditary deficits of humoral and cell-mediated immunity. Many patients with SCED have lymphocyte activation defects that remain uncharacterized. Methods: we performed genetic studies in 4 patients from 4 families of northern pre-ancestry who had clinical characteristics of SCED, including early onset of cerebral, bacterial, and fungal infections despite normal B cell and T cell counts. Genomic width homozygote mapping was used to identify a candidate region, which was found on chromosome 8. All genes within this interval were second immune cell population, signal transduction of activation, and effector function were studied.
The humoral immune response is initiated by a macrophage, which takes up an antigen, such as a bacterium or virus, and processes it. The macrophage engulfs the antigen in an endocytic vesicle and partially digests it. Class II MHC proteins bind fragments of the antigen and transport them to the cell surface for display. A cell displaying such foreign antigens on its MHC proteins is called an antigen-presenting cell. A helper T cell can bind the displayed antigen if the T cell has surface T cell receptors that correspond to a specific antigenic determinant on the displayed antigen. Once the helper T cell has docked, the antigen-presenting cell releases interleukin-1, a cytokine that activates the helper T cell. The activated T cell now produces a different cytokine, interleukin-2, as well as cell surface receptors for IL-2. IL-2 triggers helper T cells to divide. The daughter cells respond to interleukin-2 by producing a clone of cells identical to the first T cell in genetic makeup and immunologic specificity. Production of this clone completes the activation phase of the humoral immune response. In the effector phase of the humoral immune response, activated helper T cells help B cells develop into antibody-producing plasma cells. Consider a B cell which has encountered the same antigen as was recognized by the original helper T cell. The B cell processes the antigen, and class II MHC proteins transport the pieces for display on the B cell surface. Thus, the B cell becomes an antigen-presenting cell. If an activated helper T cell has the same specificity as the B cell, it can dock with the B cell and assist it by releasing cytokines. The cytokines trigger the B cell to divide, producing a clone of cells that differentiate into plasma cells. the plasma cells produce identical antibody molecules specific for the antigenic determinant that was displayed by the first B cell, and by the macrophage that activated the first helper T cell. The humoral immune system responds to every antigen in this manner, but different antigenic determinants require different specific helper T cells and B cells.
Key points. Among them, mutation in Ikbkb caused the absence of Ikbkb and a lack of T and B cell activation through their antigen receptors. Ikbkb is not necessary for development of T or B lymphocytes, but is important for their activation and for the development function of NK cells.
There are a number of types of T cells involved in specific immunity. T cells recognize their specific foreign protein or antigen only when it is presented on the surface of a body cell. One type of T cell is the cytotoxic or killer T cell. Its role is to destroy infected or cancerous body cells. In this case, a pathogen enters the cell on the left, and that cell moves foreign protein to its surface. A patrolling killer T cell recognizes and binds to that protein and releases perforin, a protein that ruptures the membrane of the infected cell. A second type of T cell is the helper T cell. One of the roles of a helper T cell is to activate B cells. At the top of the screen, a phagocytic white blood cell is presenting antigen from a pathogen it has engulfed. The B cell that responds has antibodies for the same antigen that activates the helper T cell. The T cell sends out chemical signals that affect various immune cells. These chemicals stimulate the B cell to divide and produce a clone of antibody-releasing cells.
Adaptive or acquired immunity is the system that comes online when pathogens breach surface immunity and begin to overwhelm innate immunity. It begins with the antigen-presenting cells from innate immunity, the macrophages and dendritic cells, which eat pathogens whole and break them into little protein bits called epitopes to present to T cells. B cells, meanwhile, are able to recognize entire pathogens. If there's a match between a T cell and a B cell recognizing the same pathogen, this converts the naive T cell into a helper T cell, which secretes chemicals called interleukins. These stimulate macrophages and B cells and also cause the stimulation of killer T cells. Stimulated B cells become plasma cells, which secrete antibodies, and memory B cells for a faster response. The antibodies coat the pathogens. Macrophages love coated pathogens. They also secrete complement, which forms protein rings that punch holes in pathogens and cause them to burst like water balloons. Together, this leads to successful repulsion of the pathogen attack.
Conclusions. A form of human SCED is characterized by normal lymphocyte development despite a loss of Ikbkb2 function. Ikbkb2 deficiency results in an impaired response to activation stimuli in a variety of immune cells, leading to clinical relevance, impairment of adaptive and innate immunity, Although in TT2, deficiency is lethal in mouse embryons, our observations suggest a more restricted, unique role of Ikbkb NFD, signaling its humans, funded by the German Federal Ministry of the Education in Research and others.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
The immune response involves white blood cells called lymphocytes. The most important are B and T cells. Each B and T cell is programmed to recognize and respond to one specific protein called an antigen. Different antigens are present on cell membranes. When lymphocytes encounter their specific antigens, they bind in a lock and key manner and destroy the cell. B cells respond by transforming into antibody-secreting cells. Their antibody binds to the antigen, causing its destruction. Some B cells remain in circulation carrying the memory of that antigen. T cells respond by transforming into helper, cytotoxic, or memory cells. T helper cells help B cells change into antibody-secreting cells. Without helper T cells presenting antigens to a B cell, the B cell does not respond. Cytotoxic T cells act alone without B cells. The cells destroyed by cytotoxic T cells are those infected by a virus or a cancer cell changed by mutation. T cells can become memory cells and remain in the circulation for years, ready to respond again if their antigen should appear. The first time B and T cells are exposed to a specific antigen, the response takes weeks. If re-exposure occurs, B and T memory cells respond immediately to destroy the invader. Normally, the immune response is well controlled. If not, autoimmune disease may occur and self-antigens may be attacked.